Hello and welcome to Alexander Disease Research Update, episode number 27, recorded on July 2nd, 2025. I'm Albie Messing from the Waisman Center at the University of Wisconsin, and with me today are Mel Feeney from Brigham and Women's Hospital in Boston and Natasha Snyder from the University of North Carolina, Chapel Hill. Both have been on many times before, so we can skip introductions. Before we get started, please send feedback and questions to my email, aMessing@wisc.edu. Today we're going to talk about a paper that has just come out, written by myself, Amy Waldman, and Dan Bolt, a statistician at Wisconsin, entitled Genotype-Phenotype Association for 14 GFAP Variants in Alexander Disease, which is now in press in the journal Neurology Genetics. And the key message from this paper is that even though in the past we've been able to say that some variants are predictable in their effects, we can actually do this for more than previously appreciated. First, let me address this question of definitions. What is a genotype, which is the particular sequence you have in the GFAP gene, and changes in that sequence would be these variants. And then what is phenotype? And that consists of the observable traits that an individual has. So we know that if you have one of these variants in GFAP, you will get Alexander disease, but the question is whether we can be more specific about that. In particular, will it be mild or will it be severe? What we really concentrated on in this paper were only two aspects of the phenotype. One was age of onset, which is basically the first symptom that was attributable to Alexander disease. And secondly was survival. Survival ends up being a complicated analysis. So I do want to talk a little bit more about that towards the end, about exactly what our survival curves mean or don't mean. Now, much of this information was already in the literature, but just not collected in one place. So let me just start by explaining briefly how we did it. I started out by compiling a list of all the publications on Alexander disease that contained genetic information since the initial report on genetics in 2001. And there were 364 of those papers. And then combing through those papers, we determined how many of those contained case reports, that is, descriptions of individuals with their genetic results and some description of the severity or the nature of their disease. And there were a total of 419 of those reports. And then we made an important decision. We decided that there had to be at least 7 independent reports, that is, people who were unrelated to each other, of a particular variant to be included in our analysis. Because obviously, if there's just one report, then we have no idea how predictable it is. And certainly, I would say, even with 2, we really have no idea. It was then in a gray zone between 3 and 6, but for various reasons, we just decided to set 7 as the minimum. We ended up with 10 variants out of a total of 161 that have been described, 10 variants that met this criterion for 7 or more independent reports. A couple of those actually had several different changes at the same amino acid, some of which didn't meet the criteria of 7, but we included them in some part of the study, and that's how we ended up with 14 in the title. Let me just say 2 things briefly and then open it up to Mel and Natasha. First of all, there were a few variants which I think we've known for a long time and has been stated over and over again, cause severe disease. One of them is R239, is arginine at position 239, either changing to a cysteine or a histidine caused what people referred to as severe disease. We also knew that another common variant, arginine at position 416, changing to a tryptophan, was quite variable in effects, sometimes causing mild disease, sometimes causing severe. Beyond that, we didn't really have very much reliable information all collected in one place. And now I think we can say pretty clearly that there are at least two variants that consistently cause mild disease. And there are at least three or four that cause severe disease and some that are quite variable. This paper, I think, does a good job of tabulating those results and illustrating them graphically, which will, I think, be an important resource for clinicians offering genetic counseling to people who are being given a new diagnosis of Alexander disease. So let me stop with my pitch and I'll just open it up to Natasha and Mel to grill me on what this really means.
Well, I wanted to start, maybe we could go back to what you just said about the magical number of seven. And I wondered if you could tell us a little bit more about how you arrived at seven. So for instance, why did you decide four, for instance, or five? Because, the smaller number of cases, the more potential data you could have included. So how did you really arrive at seven?
One of the reviewers of the paper asked that specific questions. The important criterion in my mind was that these people would be unrelated to each other because there are some large families with we refer to as private mutations because it really only occurred once.
So if you were just looking really in the same family, those wouldn't really be independent, was the issue.
Yeah, it would have introduced confounding genetic relatedness within a family that you don't get from people who are unrelated. And that would have been more complicated. And if you looked at the number six independent reports, some of those were, to me, possibly related to each other. If that were true, which couldn't really be determined from those papers, then they wouldn't really be six independent reports. They would have been two or three independent reports. It's too bad in a way because there were some very interesting variants at four and five, but I couldn't include them without including six. And so we ended up with this magical number of seven.
So could I just follow up on that? Because one of the really interesting things, and I don't think you really talked about it very much in the paper, but I think it might be interesting to people, is to what extent do the mutations act the same way or different ways within a family, right? So you said, okay, we don't want to be looking at the same families. We're just going to take one from each family as best we can. But what about within a family? Are they similar within a family? What do you know about that?
For the most part, they are similar. There are certainly reports of families where you've got both adult onset, for instance, and juvenile onset. But for the most part, they are consistent. And what I would really focus on actually as a good set of examples would be identical twins. And there are, I think, six identical sets of identical twins described in the literature. What's really interesting about these twins, there were three that were included in our study, but there are three others with fewer examples of that variant that weren't included. And they are very, very consistent with each other. To me, that indicates that genetics is largely driving severity of the disease, although there can certainly be environmental modifiers that influence the course. And the environmental modifiers that everyone thinks about but haven't really studied in great deal would be things like traumatic brain injury or head injury, alcohol abuse, and infection. But they've never really been studied in a large series of patients. And so it makes sense to us that they would influence the disease, largely because we think about those influencing levels of GFAP expression. In the cases of identical twins, we only took one of them, and we didn't consider the second one.
But I did think the issue of identical twins was very interesting in the paper. As you say, they seem quite similar to one another. And you've just indicated that it suggests that genetics plays a very strong role in severity. doesn't it really suggest also that things like traumatic brain injury or infection or other things are maybe less likely to play an important role?
We just don't know. I think if you look at individual case reports, there are people who have a variant, might have mild or even asymptomatic disease, and then they have one of these incidents of head injury, falling off a bike, or binge drinking of alcohol, something like that. And then they suddenly deteriorate in a much more rapid course. So that's why, people I think are certainly considering the impact of environmental factors. In one of those examples I just gave, you could also interpret that as you have let's say, a mild variant. It's actually causing no observable symptoms. You're either what you would call pre-symptomatic or just asymptomatic. And then the injury is the initiator of the period you would call clinical disease.
Well, I just wonder though, I think one of the real challenges in neurologic disease and all disease is the tendency to, ascribe a cause to something sort of in retrospect, but not
Yeah.
have that really be the cause. And I think your approach of really just asking how do identical twins look and seeing, well, they look really very similar is more of a data-driven approach as opposed to an anecdotal approach and might tend to raise the possibility that these, reporting exacerbations or initiations are, nothing more than coincidence.
And the question about twins, I think twins are really interesting. What you brought up are questions that we didn't ask or we didn't address in this paper. We deliberately took only one of the two twins, and we didn't ask how close or discrepant they were to each other here. I think that would really be an important second paper. And whether we could do it in a way that avoids privacy issues, I'm not really sure. We only know about six pairs. I'm sure there are others out there. They just haven't been published, or they're not part of the natural history study at CHOP. So that was a group of people we used to supplement the majority of the cases we analyzed, which were from publications.
So in terms of environmental factors that could help maybe with a milder course, nothing has popped up like quality of sleep or how long were they breastfed versus
Oh, you
formula,
mean
things like that.
environmental factors that improve
It
the
improves
course?
the outcome, yeah. Because I mean, I
Yeah,
think for things that worsen the outcome, there's not much you can do, but maybe
right.
could use something as an intervention that could be a lifestyle. I agree with Mel, these are really hard and anecdotal, but sometimes the parents of a child with a disease like this, they just know certain things that a doctor looking at a report wouldn't know, and things to keep an open eye for. I think
Right.
sleep is really important, and I don't know. I mean, people vary in their sleep even when they're little, and
Sleep is something that is talked about quite a bit, but usually in the context of obstructive sleep apnea. I think that's potentially different, though, than other pathways for interfering with sleep. And at a cellular level, there's some data that astrocytes are important in regulating sleep. So I think sleep is a really interesting thing that could be studied. We've tried to collect some preliminary data in the animal model, and it's just preliminary. So we need to put a lot more effort into that.
well, is it too early to get into the molecular nitty-gritty?
No, go for it.
So the many mutations in GFAP that cause Alexander disease remind me of another group of diseases called laminopathies. And these are caused by mutations in a gene called LMNA, and it's also an intermediate filament. It encodes an intermediate filament protein like GFAP. It's just located to a different compartment in the cell, which is the nucleus where our DNA is. So in the case of laminopathies, these are caused by mutations, most frequently missense point mutations in the lamin A gene that convert an amino acid to a different amino acid. And sometimes you can have a different disease caused by the same mutation, or different severity of disease. And it's been difficult to explain how you could have the same mutation causing a different disease. And I still think it's not known for Lamin, but there's been a hypothesis proposed that one type of disease cluster could be caused by structural problems with the mutation interfering with the structure of lamin, which is like deforming the nucleus, and that's pretty severe. And one example of a disease like that is a premature aging disease called Hutchinson-Gilford progeria syndrome. So because all of the nuclei in all the cells would have this problem, this is a systemic disease. Then another explanation is that some mutation maybe aren't severely impacting the structure, but they're impacting how the lamin protein interacts with other protein or chromatin or associates with the nuclear envelope. So that's more of a signaling problem. And that could explain why you only see that in certain cell types, like in the fat cells or the muscle cells, for example. And so I just wonder if with GFAP, we could have a similar problem. And the reason I started thinking about that is so the N77 site and the R79 site are both within this highly conserved motif one of intermediate filaments, the LNDR, which is in the coil one of all IF proteins. And it's known if you mess up that part, you don't form filaments. And so it seems like when you interfere with the filament structure, the formation of the filaments, that seemed to be a more severe, based on your paper, at least like with the survival data for N77, it seems like that could be consistent. The structural problems could also come from introduction of another amino acid that could be modified in some way. So the N77 to S could be introducing a new phosphorylation site. But the bottom line is that it seems like that stretch of amino acids, 76 to 79, is really sensitive, and it shows up in the disease setting. And the reason I'm thinking about like how to classify these mutations in terms of, is it a structural, is it a signaling problem? Because that could really affect how we think about targeting GFAP. And I think you and I have talked, and I agree with you that suppressing it for as many mutations as they are, and it seems like the protein doesn't do anything good, it just causes problems. So suppressing its expression should be the number one way to go about it. But if we want to try other things like stabilizing the structure or fixing protein-protein interactions, I think that's where the mutations and having some granularity in terms of what it means to have this mutation in terms of the protein and how it's behaving in the cell, that could be really helpful. So I think this study is turning a new page in how we think about doing, at least for me, it made me think more about classifying these mutations. And so I don't know what both of your ideas are about that, and if it's too early to say anything about structural versus signaling problem, how we can use this to go back to the bench now and like test these ideas.
Well, I wish we knew more about GFAP structure, what's been done so far in terms of really high resolution and modeling of intermediate filament structures has been on others, particularly Vimentin. Fortunately for us, Vimentin is also in the type 3 intermediate filament group, so it's very close to GFAP, but still that kind of really detailed structural modeling yet hasn't been done for GFAP. In the same token, practically nothing, I think, has been done about signaling roles of GFAP. So I think it's wide open. But I just want to stress that it's not just the amino acid position that matters, because I think to some degree it is the nature of the change, what the amino acid is changing to. And we don't have a lot of examples of that, but clearly in our paper, we show that R239, for instance, H, is worse than R239C. If you look at R79, which is another one that has a reasonably large number of cases to analyze, there's also a H and C comparison to be made, and it's not the same. So it's not just going from an arginine to a histidine is always worse than arginine to a cysteine. It's a combination of position and exactly what amino acid is changing to. I was just looking at another one today where it's not in the 7 or more group. It's fewer cases, but it's at position 73. So normally 73 is a methionine. If it's changed to isoleucine, it's a very mild and late onset disease. If it changes to threonine, it's a really severe disease. So it's at opposite end of the spectrum in terms of severity. Now, it's based on very few cases, so it didn't meet our criteria of 7, but that's certainly what these suggested.
I think the differences and the changes in specific residues is fascinating. And I just wonder if there's any way to take advantage of what I know you both know is the recent Nobel Prize winning discovery of computationally modeling structure. So could we computationally model the structure using something like AlphaFold and then ask, maybe Natasha's already done this and can tell us the answer, and then ask what changes in the structure, computationally determined structure, do these changes make?
That is such a good question. And so I have someone working on this now. And interestingly, if you just go through the Alexander disease causing mutations and put them in, it doesn't do much to the structure. If you introduce a post-translational modification site, we've done a few, but like serine 13, 16, 17, those are all in the head domain. Interestingly, it converts the unstructured head domain to a coil. It extends the coil region of GFAP, and it's really profound. So I think it's the post-translational modifications that have a major impact as opposed to the individual mutations.
Well, that's very interesting because if you think about other protein aggregation diseases, it often is post-translational modifications that really influence aggregation. So that's a very interesting thought in an area. So I had a question about the survival curves, and I was looking at the survival curves for patients with R79C versus R79H. And I noticed that when the patients were younger, the two curves looked very similar, but only when the patients were older did the curves move apart. And I wondered what that meant.
I'm glad you asked that question because there are some aspects of these survival curves that are really important and also easily misunderstood. So the Kaplan-Meier curves show proportion of a population that's surviving on the y-axis as a function of time. So age is on the x-axis. So as you move from left to right, you're getting older, and you're showing with these curves how many in the population are still alive. But the real advantage of Kaplan-Meier curves was that you could use data that's incomplete in many ways. The reality in the Alexander disease group is that we know that people were alive at a certain point. For instance, they came in for a clinic visit, but some of those we stay in touch with and we know they're alive as of today, but others are lost to follow-up. The advantage of the Kaplan-Meier curve analysis is that we could take advantage of what we do know about them to compare one curve versus another. So in the case of the R79C and H, that's a perfect example of this because they are very close early on. They seem to diverge, but that's because many of them have been lost to follow-up, and we have very few after the age of 20 or so. And so statistically, those two curves are not different. In the case of R239, C, and H, those curves are different. And in that case, H is worse than C.
And that's a case where even early on, you start to see the difference. And you're saying that's because you have many more patients really in that phase of the curve.
Right. Actually, we had an interesting question submitted by patient family community about the Kaplan-Meier curves. This one had to do with N77S, if anyone wants to look at that curve. But this is an example of a variant that has fairly early onset, and the Kaplan-Meier curve shows a relatively steep decline. But then it continues out to the age of 30, and that's where it stops. What it means is that there is at least one person who's still alive at the age of 30. So the question was, with a variant like N77S, where there's such a marked disparity between many of the patients who have severe disease and a sharp decline, but some who's living a very long time.
Yeah, I think the question of adaptation, because somehow that patient that lived to 30 or beyond, something about their biology has allowed them to adapt to the presence of that particular mutation and overcome maybe a critical juncture in the development and progression that their cells just learn how to live with it. That would be so interesting to look at in future studies.
Well, let me say about future studies. The editor of the journal implied that the real value is in these large case studies, lots of individuals. But I think this actually brings up a situation where there's really continued interest and need to study individuals and not necessarily large case studies. And so that N77 patient who's living at age 30 is one example of that. The other thing for future studies is that, we set this criterion of 7, which is arbitrary, and didn't really pay that much attention to the ones below that. But as time goes on, hopefully, as more patients are published, we'll have more case reports that will allow certain additional variants to rise above that threshold, and then we could include them in this type of analysis.
I wonder what role you think whole genome sequencing might have to help us understand, differences in families and mutations in general, and maybe even more specifically for these very interesting cases of late onset or long survival.
I think it's great that whole genome sequencing is becoming more and more available, even though the analysis is much more complicated. I think basically what it's going to give us are a lot of candidate modifiers that we think in other genes that might influence the course of Alexander disease. I think there are going to be a lot to choose from, and the challenge will be to find practical model systems or assays to use to evaluate their effects.
And I guess the other sort of thing that you didn't see, and I wondered if you could comment on it, is you didn't see an effect of sex. And was that expected? Had people thought in the past that there might be a worsening in one sex versus another?
So we go back and forth on this. The early literature, which is to say, even up through maybe 2005 or so, suggested that boys were more severely affected than girls. And of course, the early literature on Alexander disease was dominated by early onset cases. And so it was a little bit skewed in that way. After 2005, and certainly by 2010 or 11, when much larger series of cases were being analyzed, the sex disparity went away. And so I think our study is reinforcing that finding that there's really no difference in the sexes. You go back to the rodents, though, and in the both mice and the rats, we find hints of sex differences, and we don't want to ignore them, certainly, if they're detectable, but there hasn't yet been a way to translate those to the human. So I think the story in human right now is consistent that there's no sex effect.
And I had a very general question for you, Albie. When I was reading over this paper in detail, I noticed that there were a lot of details that you guys had to think about. And I just wondered from your perspective, what was the most challenging part of the study?
I think just deciding how to define age of onset was challenging, both in our own minds and in terms of inferring what was trying to be communicated in these publications. It's easy to define age of onset, for instance, as a seizure. Everybody remembers the date of the first seizure. A seizure is something that prompts a medical attention. You don't sit at home with a seizure. You go see a doctor, you try to get to the source of that. So everybody remembers a seizure, and that's a discrete age of onset. But then there's this whole idea of developmental delays in acquisition of motor milestones or cognitive milestones in language. And of course, there's a lot of variability in normal development. And so trying to interpret what a developmental delay means in terms of exact age of onset can be challenging. We had certain terms that we saw in the literature, such as infancy. For statistical reasons, we needed to convert a term like that into a number. And so we explained what we did, for better or worse. And it had to be a number for statistics. And I don't think it fundamentally skewed or distorted the results. But for instance, infancy, we interpreted as 1. It might have actually been earlier than that, but it was 1. And so making those decisions was sometimes difficult. Deciding on 7 as a criterion was painful because I thought there was so much worthwhile data in reports with fewer independent case reports. But as I said, there were problems with those at 6. And without being able to deal with those problems, we couldn't go below that.
Well, I think it's very exciting work. I think it's fascinating. And with Natasha, I'm really excited about the potential to understand molecularly how these various mutations work. And I think there's a lot of fodder for future experimentation there.
All right, that's all for today's episode of Alexander Disease Research Update. Thanks for listening, and thank you to Mel and Natasha for joining me today. Our theme song was written by Charlie Allenson, and support for these podcasts has come from the Barrett Riddle family. I'm Albie Messing. See you next time.
Thank you, Albie.